Welcome to this course on biosynthesis of macromolecules. Macromolecules are the building blocks of life. They are large, complex molecules made up of smaller units, and they perform vital roles in the structure, storage, communication, and regulation of biological systems. In this first module, we will discuss the structure and function of the major classes of macromolecules. We will explore storage and structural polysaccharides, mucopolysaccharides, glycoproteins, and complex lipids. By the end, you should have a clear understanding of how these molecules are built, why they are important, and the fundamental processes involved in their biosynthesis. Section 1: Overview of macromolecules. There are four major classes of biological macromolecules. Carbohydrates, which include polysaccharides, serving as energy storage and structural components. Lipids, which form membranes, store energy, and act as signaling molecules. Proteins, which serve as enzymes, structural elements, and regulators. Nucleic acids, DNA and RNA, which store and transmit genetic information. In biosynthesis, cells must carefully assemble these molecules using specific enzymes and pathways, ensuring both precision and efficiency. Section 2: Storage and structural polysaccharides. Polysaccharides are long chains of monosaccharide units joined by glycosidic bonds. Storage polysaccharides include starch in plants, composed of amylose and amylopectin, serving as the main energy reserve. Glycogen in animals, highly branched and stored in liver and muscle, providing quick access to glucose when energy is needed. Structural polysaccharides include cellulose in plant cell walls, linear chains of glucose linked by beta-1, 4 bonds, giving rigidity and strength. Chitin in insect exoskeletons and fungal cell walls, similar to cellulose but with N-acetylglucosamine subunits. Biosynthesis of these polysaccharides involves enzymes such as glycosyltransferases, which catalyze the transfer of sugar residues from activated nucleotide sugars like UDP glucose to growing chains. Section 3: Mucopolysaccharides, glycosaminoglycans. Mucopolysaccharides, also called glycosaminoglycans or GAGs, are long unbranched polysaccharides consisting of repeating disaccharide units. Examples include hyaluronic acid, provides lubrication in joints and forms part of the extracellular matrix. Chondroitin sulfate, found in cartilage, providing resistance to compression. Heparin, an anticoagulant that prevents blood clotting. These molecules are highly negatively charged due to sulfate and carboxyl groups, making them capable of binding large amounts of water and forming gels. The biosynthesis of GAGs occurs in the Golgi apparatus, where alternating sugar residues and sulfate groups are added step by step. Section 4: Glycoproteins. Glycoproteins are proteins covalently bonded to carbohydrate chains. They play critical roles in cell-cell recognition, signaling, and immune responses. N-linked glycoproteins, where carbohydrates are attached to the nitrogen atom of asparagine side chains. O-linked glycoproteins, where sugars attach to the oxygen atom of serine or threonine residues. Examples include antibodies, hormone receptors, and components of the extracellular matrix. The biosynthesis of glycoproteins begins in the endoplasmic reticulum, where a preassembled oligosaccharide is attached to the protein, followed by further processing and modification in the Golgi. Section 5: Complex lipids and lipoproteins. Lipids are a diverse group of macromolecules, but complex lipids include those that are conjugated with other biomolecules. Phospholipids, essential for forming cellular membranes. Glycolipids, lipids with carbohydrate groups, important in cell recognition. Sphingolipids, found in nerve tissue, crucial for signaling and insulation. Lipoproteins are assemblies of lipids and proteins that transport fats in the bloodstream. They include chylomicrons, carrying dietary triglycerides. LDL, low-density lipoprotein, often called bad cholesterol, delivering cholesterol to tissues. HDL, high-density lipoprotein, good cholesterol, removing excess cholesterol from tissues. The biosynthesis of complex lipids involves pathways in the endoplasmic reticulum and Golgi, requiring specific enzymes for fatty acid elongation, desaturation, and conjugation with proteins or sugars. In this module, we explored the structure and function of key macromolecules, polysaccharides, glycosaminoglycans, glycoproteins, complex lipids, and lipoproteins. We also discussed the basic principles of their biosynthesis and their functional roles in living systems.